Have you ever wondered how that apple in your hand got so perfectly crisp and sweet? Or how your strawberries can last for a week in the fridge without turning to mush? Well, that's no accident. For thousands of years, we've been on this quest to make our food better. And it all comes down to solving one giant fundamental challenge.
Yeah, this question, it's one of the most important ones we face, right? We need crops that can fight off diseases, handle weird weather from climate change, and pack more nutrition into every bite. So, how have we been tackling this traditionally? For the longest time, we basically had two main tools in our toolbox. You've got your classical breeding, the old school way of crossing plants and hoping for the best. It works, for sure, but man, is it slow. And it relies on a ton of luck. On the other end, you have genetic modification, or GMOs. Super precise, right? But it also comes with a whole lot of public debate and regulatory hoops to jump through.
So you can see the problem, right? There was this massive gap in the middle. What scientists really needed was a third option, something that could speed up the natural process of genetic variation, but without introducing foreign DNA and all the baggage that comes with it. And that need for innovation led to an incredibly powerful technique called tilling. Turns out, it's that clever middle ground everyone was looking for. Okay, so tilling stands for Targeting Induced Local Lesions in Genomes. Whoa, that's a mouthful, I know, but the idea behind it is actually super elegant. It takes an old-school tool, chemical mutagenesis, which just speeds up how nature creates random little genetic typos, and mashes it up with brand-new, super-fast DNA screening. It lets scientists find one single beneficial typo in a plant's entire genetic library. And it does this by kind of flipping genetics upside down. You see, scientists used to rely on what's called forward genetics. You'd find a plant with some cool trait, like being really tough against drought, and then you'd spend forever trying to figure out which gene was responsible. Tilling does the opposite. It's reverse genetics. You start with a gene you already know is important, and then you go hunting for mutations in that specific gene to see what cool new traits pop up. This library analogy here just nails it. Instead of randomly finding a cool book, it's like you already know the title you want and you use a high-tech scanner to find the one copy in the entire library with a tiny interesting typo.
So what does that high-tech scanning process actually look like in the lab? Let's walk through it.
So here's the game plan. First, scientists zap thousands of seeds with a chemical that creates those tiny random DNA typos. Then they grow up those seeds and let them produce offspring, which locks in those mutations. Now here's where it gets really efficient. Instead of checking every single plant, they just take DNA from thousands of them and mix it all together into one big pool. Then, using PCR, they make millions of copies of just the one gene they're interested in. And this is where the real magic happens. Okay, so how on earth do you find one single mutated gene swimming in a giant pool of normal ones? It's genius. They heat up the DNA, which makes the double helix split into two separate strands. As it cools down, the strands find their partners and zip back up. But if a mutated strand pairs with a normal one, it doesn't zip up perfectly. Right where that little typo is, it creates a tiny bubble, or a mismatch. And to find that tiny bubble, they use a special enzyme. And get this, it's often an enzyme called cell-I, which comes from, of all things, celery. This enzyme is like a tiny pair of molecular scissors that's programmed to find that exact mismatch and snip the DNA right there. That snip is like raising a big red flag that says, hey, mutation over here. From there, they can work backwards to find the one individual plant that has the prize. I know this all sounds pretty technical and deep in the weeds of genetics, but the results are actually changing what you see in the grocery store and what farmers plant in their fields. I mean, tilling has already delivered some incredible wins. We are talking about tomatoes that stay fresh on the counter for way longer, wheat that can fight off a nasty fungal disease, and even peanuts with lower levels of allergens. It's also been used to boost the fiber in wheat and make the nutrients in rice easier for our bodies to absorb. The point is, this isn't some far-off theory. This tech is actively making our food better right now. And tilling is part of a much bigger story. Using mutation to improve crops has been a pillar of agriculture for a long, long time. We're talking over 3,300 new and improved crop varieties released worldwide. Techniques like tilling are just putting that whole process on hyperdrive, making it way faster and more precise than ever. This quote really just nails it, doesn't it? It shows the two sides of tilling's power. It's an amazing tool for scientists in the lab trying to figure out what genes actually do. That's the functional genomics part, but it's also an incredibly practical tool for plant breeders who are out there trying to make better food for all of us.
And of course, like any awesome piece of technology, tilling hasn't just been sitting still. It's evolved. And now there are new, even more powerful versions out there.
So one of the first big spin-offs is something called eco-tilling. Now remember how regular tilling finds mutations that we made with chemicals? Well, eco-tilling uses the exact same detection method, but instead it looks for natural genetic differences that already exist out in the wild. It's like a super fast way to scan Mother Nature's own genetic experiments to find useful traits.
And the next big leap is called tilling by sequencing, or T by S. This upgrade totally gets rid of the celery enzyme. Instead of looking for that little snip in the DNA, it just uses next-generation sequencing to read the DNA sequence directly. This lets scientists find the mutation just by reading the code, which makes the whole thing faster, more precise, and lets them screen an absolutely massive number of plants all at once. It's a real game changer.
So in today's world, with powerful tools like CRISPR out there, you might be asking, why is tilling still so important? Well, first off, it's non-GMO, which is a huge deal for public acceptance. Plus, you can use it on almost any plant species. And once you create a tilling library of mutated seeds, it's there forever, a permanent resource. But maybe the coolest thing is that it creates what's called an allelic series. Think of it like having a dimmer switch instead of a simple on-off switch for a gene. You get a whole range of mutations, from mild to strong, which lets breeders fine-tune a trait perfectly.
When you think about it, tilling really is this perfect bridge. It takes the randomness of old-school breeding but uses the precision of modern genomics to find the needle in the haystack. It's a powerful reminder that sometimes the best solutions are a smart blend of old ideas and new technology. So the next time you bite into an extra juicy tomato, just think, you might be enjoying the result of a single, perfectly targeted typo.